Stop right there. You're over your calorie count. In the name of caloric restriction, I taught this doing this.
No!
So we all know about counting calories. We've all seen fad diets come and go. One day, fats are the enemy. Another day, carbs are the source of all weight gain. And eventually, we're drinking disgusting juice cleanses in the name of detoxing. This is all for the sake of losing weight. But did you know that caloric restriction could actually improve your lifespan? And we're not just talking about fad diets that don't work. We're talking about limiting your calories while still having enough nutrients for your body. Caloric restriction is one of the few anti-aging interventions that has definitive scientific evidence to back it up. But how strong is this evidence? Could it actually help humans increase their lifespan? Let's find out together.
So far, what we know about calorie restriction, or CR, is that it can slow biological aging, which is correlated with increased longevity. CR has also been shown to improve health span, the period of lifespan in good health, by decreasing the likelihood of various cancers, including hepatoma, lymphoma, and lung cancer in animal studies. However, there are caveats to CR, such as decreased body temperature, decreased insulin secretion, and increased irritability. In further support of its effects on lifespan, CR is linked to several evolutionary theories of aging and underlying pathways. One example is the oxidative damage theory, as reduced metabolic activity leads to lower production of reactive oxygen species, decreasing oxidative stress, and potentially increasing longevity. Also, CR affects insulin signaling by lowering insulin levels, which is associated with pathways such as FOXO and DAF2 that inhibit growth-related genes and activate stress response and longevity genes. Evidence for CR has been strongest in simpler organisms like worms, flies, and mice, where numerous studies have shown positive results. Today, we'll take a closer look at some of these studies, moving from mice to primates to humans.
In this first study, researchers studied male mice to observe whether CR, fasting duration, and when they ate affected lifespan. Here's what they found. CR without fasting or timing was enough to extend median lifespan by 10.5% more than the freely eating mice. CR paired with fasting during the day resulted in mice living, on average, 18.9 to 21.2% longer. Lastly, CR paired with fasting and nighttime feeding in alignment with the mice's natural body clock extended median lifespan by 33.6 to 34.8%. Beyond direct changes to median lifespan, caloric-restricted groups also had improved hormonal profiles, insulin sensitivity, and glucose homeostasis, which may indirectly influence lifespan. While the results appear to demonstrate that CR, in conjunction with feeding time and fasting, mediated lifespan extension, there are some important caveats. Firstly, the authors measured median lifespan, or how long the average mouse lived, as opposed to maximum lifespan, or the longest-lived mouse, so the authors are not directly demonstrating an extension of longevity. Additionally, there may be species-specific or sex-specific responses to circadian alignment, as well as differences in human sleep and activity patterns. So while the results are compelling, we cannot extrapolate these findings onto humans until more research is conducted.
Few studies have examined the health and longevity effects of caloric restriction in primates, but the results from this 20-year longitudinal study took a major step in addressing this knowledge gap. 46 male and 30 female rhesus monkeys were randomly assigned to either a control or CR group, characterized by a 30% reduction in caloric intake. The results indicate that CR's effect on age-related mortality is significant, with 37% of control monkeys dying from age-related diseases compared to only 13% of CR monkeys. The effects of CR on health span were even more significant. CR monkeys had improved glucose homeostasis and diabetes prevention. Additionally, neoplasia and cardiovascular disease incidence were both reduced by 50%. The group reached a conclusion that CR monkeys appeared to be biologically younger than control ones. These results suggest that, given the obvious parallels between rhesus monkeys and humans, the beneficial effects of CR may also occur in humans. It is worth noting, however, that 2 other CR studies in monkeys had less overt results. Thus, it may be difficult to translate these findings directly into practical solutions for humans.
In this paper, they used data from the NIH-funded Calorie Trial, a randomized trial study of about 220 healthy non-obese adults. They compared a control group to a 25% calorie restriction group over 2 years, checking in at 12 and 24 months. The results showed that after 1 year, the control group aged about 0.71 biological years, while the calorie restriction group only aged about 0.11, based on biomarkers like blood pressure, cholesterol, and inflammation. And even after accounting for weight loss, the slowing of aging was still significant. Interestingly, some people did less than 10% calorie restriction while others did more, but even those with less than 10% still showed significantly slowed aging, and doing more did not add a huge extra benefit. That said, there are some caveats. They're measuring aging using biomarkers, not actual lifespan, and there was a wide range of adherence to the calorie restriction regimen, so it's hard to generalize. But overall, it is still a very promising result. Then the next study specifically looks at reduced inflammation as a possible mechanism of slowed aging. The researchers followed a similar group of 218 healthy participants divided into a control and 25% calorie restricted group over 2 years. They found that by around 2 years, CRP dropped about 40% and leptin about 50%. CRP is a protein made by the liver that goes up when there's inflammation, and leptin is a hormone that helps regulate energy balance and is also linked to inflammation. At the end of the study, they tested whether immune function was maintained by measuring vaccine response using antibody levels. The results showed that inflammation decreased without compromising immune function. This study has similar limitations in that it used biomarkers and not a very diverse pool of participants. Both studies were also only over 2 years and do not show long-term outcomes. However, these preliminary results in humans show promising outcomes for applying caloric restriction.
Many unbearable hours later.
So with all the evidence we've looked at, what can we say?
You can see that healthspan and lifespan is increased with these mice and monkey studies we talked about. The mice and monkey are mice and monkeys. To find out if caloric restriction is right for us, we have to look at human studies. The one we discussed has promising implications, but there are a lot of confounding variables to consider. Human studies are difficult because it's hard to have a diverse sampling population, and each person is an individual. They have their genetics, their predisposed conditions, and other lifestyle choices outside of the consumption of food. They can't be kept in a monitored cage like lab animals are, and won't follow diets as strictly. Despite this, there's still promise for using caloric restriction as an anti-aging intervention, especially with the possible added benefit from intermittent fasting. We believe there's benefit in trying it, but in moderation. Start small when you cut back, and don't be too extreme in your pursuit for living a longer life. After all, is life really still fun if you cut out all the food you love?
Alright, I guess you can have your food back.